Nice. Well, thank you, everyone. Welcome today for our webinar, a joint Neo4j Elsevier webinar, which is in two parts and around the topic of the protein regulatory networks of COVID-19 and the knowledge graph which was created by Elsevier. My name is Anthony Flynn, and I'm the sales director for the UK and Ireland here at Neo4j. I'm very pleased today to be joined by Helena Deus, who's now at consulting firm ZS Associates, but who was at Elsevier and worked on the COVID-19 knowledge graph project, which she'll be presenting today. And also by Shobhna Srivastava, who's the director of engineering at Elsevier, and who will be presenting on the research citation network, which uses Neo4j. So Shobhna and Lena will be presenting these different use cases, and the connection between them is that the team working on the COVID-19 project was inspired by the technology used by Shobhna's team on their citation graph project. So without further ado, I will go ahead and hand across to Lena. Thank you, Lena.
Thank you, Anthony. All right. Hey, everyone. So today I'm going to talk to you about the protein regulatory networks in COVID-19 and how you can access them using a knowledge graph. So in March 2020, novel coronavirus, as you know, called SARS-CoV-2, took the world by surprise. It started in China, but it could have started anywhere, really. This novel coronavirus, as soon as it jumped into a human being, had at its disposal a deeply interconnected world that humans had created. It was able to very quickly use our ventilation systems, use our densely populated cities to quickly jump from person to person, virtually undetected. And in July 2020, over 11 million people have been infected with SARS-CoV-2, and the virus is responsible for 500,000 people dying in just under 6 months. And these numbers are in spite of confinement and social distancing, which have certainly achieved the intended effects of ensuring ICUs have sufficient capacity. Now, you compare these numbers against the 80 million that died of the Spanish flu, and maybe it doesn't look so bad. But remember one thing, the way humanity got rid of the Spanish flu was by overhauling the sanitation system. Now, in spite of very good sanitation, we still have 500,000 people dying off of the coronavirus. So it is my strong belief that in order to actually avoid a new pandemic like this in the future, we really do need to overhaul our entire healthcare system, not just in the US, but all around the world. So back in March, when Elsevier noticed this, one of the things we noticed, and I was working at Elsevier back then, was that the world turned its attention to scientists and healthcare workers. Can you help us? People would ask. Can you make this go away? Can you protect us? And the three most important things for scientists and healthcare workers to work on are not just back then, but we're still in this situation. Drug repurpose and vaccine repurpose, because it takes years for a new drug or a new vaccine to get approved by the FDA. We can't wait that long. And so we need to be able to find drugs that have already been shown to be safe, that can be repurposed for treating COVID-19. The second most important thing is to scale up testing. As you know, knowing exactly who is infected and who they might have infected is step number one towards slowing the spread of the virus. So if we know who's infected, then we can carry out contact tracing and know exactly where to look for the next surge of the virus. And the final thing that we really need is reliable information. So when this whole thing started, the doctors started sharing information on social media. There was not a lot of medical information, reliable medical information available. And as you know, there's a lot of things on social media that are unreliable. So being able to help doctors and healthcare workers separate the good information from the bad information, the information that they can rely on to make treatment decisions from information that is not as reliable, became pretty crucial. Not only that, the policymakers also need the ability to use good reliable information to make policy decisions. So even though we've been at this for months now, what I'm showing you here is actually fairly recent from since the end of June. we've only had one drug that has been FDA approved, that's remdesivir. Even remdesivir has had an accelerated approval because the drug is still going through clinical trials. So as you know, in order for the drug to be approved for human consumption by the FDA, it always must go through clinical trials. And so as far as vaccines go, those are even further behind. So the top 5 more promising vaccines right now being tested are BCG vaccines. So the vaccine that has been designed specifically for SARS-CoV is even further behind than all of the other attempts to use already existing vaccines to prevent the spread of the virus. All right, so with that in mind, what can you do to help COVID-19 scientists and healthcare workers move us out of this mess faster than what the pace at which we're moving now? So before I actually dive into the knowledge gap, I feel like I need to give you a bit of background on the biology. Because my guess is that most of the audience is made up of data scientists and computer science, and you've had limited exposure to biomedical knowledge. So if you are a biologist, please bear with me while I go through the biology presentation. All right, so the first thing that I want to highlight is you'll hear several terms related to COVID-19. So COVID-19 is the name of the disease, whereas SARS-CoV-2 or 2019 nCoV, this is the name of the virus itself. And I want you to think about COVID-19 is to AIDS, SARS-CoV-2 is to HIV, right? So they're related, but one is the disease and one is the virus that causes the disease. And then coronavirus is actually not one virus, it's a family of viruses or a class of viruses. And if you remember MERS and SARS, they're also caused by coronaviruses. Now, since a lot of research has been done on MERS and SARS, the knowledge graph that I'm going to tell you about is actually going to be mostly focused on what scientists have learned about MERS and SARS, even though there's a little bit of COVID-19 there as well. And before we jump deeper into more biology, I want you to keep this picture in your mind. A virus, a human virus, is actually very much like a computer virus. It's not a cell, actually. It does not have its own operating system, just like a computer virus doesn't have its own operating system. In order to make copies, in either case, a virus needs to use an operating system. In the case of computers, it's your laptop, but in the case of human virus, they need to use the DNA of the cell. They need to use the machinery that exists inside the cell to make copies of its own DNA. Right? So just like the computer virus, in order to get into your cells, into the DNA, human virus, the coronavirus, needs to first go through your skin, right? And that's the firewall that we all have that protects us from malicious virus trying to get in. Once it manages to get through, it needs to fight the immune system, and that's your antivirus. And most of us do not have a signature for this coronavirus. And so this is why the coronavirus is such a serious threat to our well-being, because even though nowadays our immune system can fight diseases like the flu and colds and whatnot, we are totally unprepared. Our immune system is totally unprepared to deal with coronavirus. And then the virus needs to get through the cell membrane. So even if it manages to evade the immune system, it still needs to get into the cells. And my equivalent metaphor for this in the computer virus is it needs to get root access. It needs to be trusted by the cell before it can actually do anything with the machinery of the cell. And finally, once it's in, it can install itself in your DNA, just like a computer virus installs itself in an operating system. And from that moment on, they've got free range to make as many copies of themselves as they want. And that's usually what happens. So a computer virus will try to replicate itself and send itself to other computers, whereas a human virus will replicate itself and send itself to other cells. So the whole purpose of the virus is to make more copies of the virus. That's all it can do. That's all it knows how to do. But how does the virus actually physically infect people? So what you're seeing here is your lungs, but blown up. So your lungs are made up of these little sacs, the alveoli. And in a healthy lung, what you see is these alveoli, these little sacs are full of air. Now, when the coronavirus is able to enter these sacs and to invade the cells that make up the sac, what's going to happen is your immune system, it can't exactly recognize coronavirus, but it can tell that there's something foreign there. So what the immune system will start doing is it will start recruiting the military, basically. You've got these natural killer cells or NK cells that will recognize foreign objects, whatever they are, and it'll start launching toxins, right? It'll start trying to kill these foreign things. And when the immune system does that, it doesn't just kill the virus, it also kills normal cells. And what you see in the third little image from the top, the third sac, what you see here is that the immune system actually starts invading the sac, and the membrane of the sac becomes more permeable. So you start seeing fluid enter the alveoli, and of course, with fluid, no air can be transferred into the bloodstream. Once this is very severe, this is when the patients need to go on the ventilator. So their sacs are completely filled with fluid. The immune system continues to try to kill whatever foreign agent. And this, of course, mixed up with now dead cells that were killed by the actions of the immune system itself. This is why when you hear in the news patients don't die of the virus, they die of cytokine storm. This is what's happening. The immune system is going berserk because it can't figure out what it is that it needs to fight, and so it'll just kill anything in sight in order to try to stop the virus. But how is it that our immune system can essentially kill us, right? It shouldn't it shouldn't kill us. We should actually be able to trust our immune system. So this is what's going on. We actually don't have one immune system, we have two. The immune system is made of T cells, which are the ones that I call them the SWAT team, right? They're the snipers. The T cells know exactly who to kill and know how to make a very targeted kill. Whereas the NK cells, the natural killer cells, they're usually the first responders, right? They're the boots on the ground, and their attacks are not very directed. But what typically happens when we do have T cells that can recognize foreign invaders, the T cells will tell the natural killer cells, okay, it's my turn, just stop doing what you're doing and let me take over from here. But what's happened with the coronavirus is, because there's no adaptive response, because the T cells never come, the innate response, the response from the natural killer cells will just keep going up. So they don't really get that signal saying, stand down, it's time for the professionals to take over. So that's what's happening with our immune system. This is why patients are actually dying by suffocating in their own immune system. All right, so with that in mind, and I gave you this background so that we can start thinking about strategies we could actually use to stop the virus, or to at least stop this highly reactive immune system from killing the patient. If you're curious about what the virus is made of, if you're curious about understanding this enemy a little better, there's this fabulous article in the New York Times called Bad News Wrapped in Protein. And they go over all of the proteins that have been identified in this viral DNA. Most of them are, we kind of know what they're doing, because we can take the protein structure and compare it against proteins we already know. And that's purely computational work, which is very fun to work on. And for example, there's a couple of proteins they highlight. This NSP10, which essentially camouflages the viral DNA so that the cell thinks that it's its own DNA instead of viral DNA. And then there's this NSP1, which is a cellular saboteur. So what this NSP1 actually is doing, it's convincing the cell that it should stop doing what it's doing, right? So the cell is normally busy going about its business in order to survive. It's, getting oxygen and generating energy, etc. And this NSP1 will actually tell the cell, hey, stop doing what you're doing. It's time to go make copies. So we know a few of these proteins, and every one of these 16 proteins, we know exactly how many there are, is a possible target for us to go after in order to actually stop the virus from causing the devastation that it causes in the human body. All right, one last thing I want to tell you. So we talked about the immune system. We talked about the 16 proteins that make up the virus. The way the virus enters the cell, and I want to focus on this detail because a lot of therapies have been focusing on ACE2. The virus actually binds or attaches itself to this ACE2 protein, which lives at the surface of every cell. And basically, ACE2 is obviously it did not evolve to let viruses in, obviously. ACE2 is a protein that's responsible for blood pressure, for maintaining a healthy blood pressure. And so when the virus uses ACE2, it basically forces ACE2 to think of the virus as a piece of food. And so ACE2 will cause the virus to be internalized into the cell. So another mechanism that can happen is, still with ACE2, the virus can actually also convince this other protein called TMPRSS2 to activate ACE2 so that it doesn't actually get into the cell via bubble. There's a much easier route for a virus when it manages to do this, which is only the DNA gets injected, or the RNA, the genetic code, gets injected into the cell. And so when the cell sees that there's a piece of genetic code, it will almost always read that piece of genetic code and start making copies and start generating whatever that genetic code is encoding for. All right, so remember that ACE2 is the mechanism that the virus uses to enter the cell. Because now we're actually going to query the knowledge graph, and ACE2 is going to play a big role. So you can, either take a snapshot, or I think everyone's going to paste the URL and the username and password in the chat box, so you guys can follow along if you like. I'll stay there for a second so that you guys can take your snapshot if you want to. All right, so moving on. All right, so the first thing we're going to try is, and I'm only going to give you snapshots, so you're going to have to imagine the rest. When you enter the browser, when you enter Neo4j, there is a little icon that looks like a database. If you click on that, you'll see all of the types of things we have in there. And what I'm going to query for is a drug. I'm going to just get a list of drugs. And I'm limiting my query to 3 so that, it doesn't look too crowded on the screen. And what you see is just, 3 bubbles show up, 3 blue bubbles show up. The drugs we have in there are also small molecules. So there are other types of drugs. But one thing that you'll see if you go to the text tab is that we also have all sorts of identifiers that you can use to look for those same drugs in other databases. You can bring data from those other databases in, and you can actually start building or expanding the graph to include other attributes about these drugs. But now let's just take one of these drugs and I call it explode it. Let's look at all of the relationships that drug has with other entities, right? So the drug I'm picking here is the Eprozart something. And what you're seeing here, all the pink bubbles, they are proteins. And proteins, as I mentioned before, they're kind of like the processes in the cell, right? They get things done, and they can be created and destroyed. And ACE2 is one of those proteins. As you can see here, there's a connection, there's a relationship between the drug I selected and ACE2. Now, the types of relationships that we care about, at least for understanding how we can actually force the virus to not enter the cell or revert the effects that the virus is having in the cell is by causing more of the protein or less of the protein. So when we use a drug to create more of a protein, we say it's being upregulated, right? So we say that the regulation is positive, and you can see one of the relationships here is the regulation process positive. And we can also do something else with the drugs. We can cause the regulation, we can cause the quantity of the protein to go down. And when you do that, it's called a downregulation or a regulation negative. Now, because, as I told you before, ACE2 is the protein that the virus uses to enter the cell, it's logical to think that if we lower the concentration of ACE2, then the virus will have less ways to enter the cell, and therefore it's possible that we will give our immune system enough time to learn how to fight the virus and to actually avoid getting sick from the virus. So if we then look into ACE2, what I'm showing you here are basically just some properties of the protein of ACE2. And one of the things you'll see here is that we have this KEGG ID. And I picked KEGG because I know it as it's a pathway database. There's other databases in there that you can link out to and query for information. But I thought this one was interesting. I'm going to show you why. So as you can see, the query I'm doing here is a down regulation. I'm finding drugs that cause less of that protein, and the protein I'm selecting is ACE2. And one of the identifiers I'm finding over here associated with the protein is this KEGG ID. Now, here's the thing I also told you. ACE2 is responsible for maintaining a healthy blood pressure. So if I just cause ACE2 numbers to go down, we need to somehow compensate for that, right? Because you don't want to fix one problem by creating another one. So if I look at that identifier on KEGG, which is a database you can find on the web, one of the things that KEGG is telling me is that ACE2 is involved in all of these mechanisms. And this diagram I'm showing you here, this double line, it just means the membrane of the cell, and these are just regulatory connections between proteins inside the cell. And as you can see, ACE2 is involved in this pathway to regulate blood pressure, but it's also involved in this pathway and this pathway. So if we cause ACE2 to go down by using a drug that causes its regulation to go down, we need to bring something back up that compensates for the fact that ACE2 is now downregulated. And so by looking at this diagram, I can see there is a path here, the T-HOP, NLM, MME path, that can actually cause can still help with vasodilation and, help with blood pressure control, called MAS1. So my logic is, if I downregulate ACE2, I want to be bringing MAS1 up, so that it compensates, so that the cells can continue to compensate with blood pressure, even though we are going to deplete them of ACE2, right? So I go back to Neo4j, and I can look for MAS1, and in fact, we can see, and again, there's a lot more drugs than this. I'm just limiting my query to only 3. But as you can see, there are drugs that can positively regulate MAS1. Right? So this gives you a very strong hypothesis for how you could combine drugs to actually both prevent the virus from entering the cell, while at the same time not causing problems in blood pressure. So you can then take one of the drugs. I selected N-acetylcysteine. And another thing you may want to do, just to kind of complete your hypothesis and get a good sense of what this drug might do in the cell, is look at everything else that this drug will upregulate or downregulate. Because the more impact a drug has in a cellular machinery, the more likely it is that there's going to be some sort of adverse side effect. So picking the right drug often means picking the one that is very specific to the thing that you want to regulate. So in our case, picking the drug that's very specific to bringing the expression of MAS1 up, but not touching any other proteins or not affecting any other proteins or minimizing the number of proteins it affects otherwise. And that's it. That's all I have for you. I think we're now going to have Shobhna give us a talk about the citation network.
Thank you very much, Lena. Yes, fascinating conversation. I was struck, actually, I'm surprised hearing your presentation, just how much overlap there is with your analogy of hacking and cyber attacks, and the idea that the virus essentially infiltrates the cell and then needs super user privilege to do its evil work. One of the other hotspot areas for graph databases is in the area of cyber security. We have who use Neo4j to model the different kinds of attacks that hackers might launch on their IT systems. And the common theme seems to be that you want to link together information, quite different kinds of information that haven't been linked together before using a graph. So in that case, it's information about their firewalls and the different access privileges, but also what kind of applications are running behind the firewall and what kind of attacks they might experience. So was that ability to link together different kinds of data, was that an important part of the value that you found from Neo4j, from the graph database?
Yeah, absolutely. But also this ability to walk the graph. The ability to just make jump several hops from a drug to a protein to a combination of proteins back to drugs. I think that is a very powerful capability that graphs provide.
Fantastic. Thank you very much then, Lena. I'll ask Shobhna to start her presentation then. Shobhna, over to you.
Hey everyone, I will go into not the science part of it, but more on the technical and the choices that we did to use Neo4j. This is not directly in reference to the COVID-19 work that was done by Lena and a couple of other folks. This is specifically the use case that we started looking at for Neo4j was enhancing search results using some sort of a graph-based technology. So Elsevier as a business is an information and analytics business in science and health domain. The product that I'm going to specifically focus on, for which we evaluated graph databases, is Scopus. And if you go to the Scopus website, it's listed there. You will see that it is listed as expertly curated abstract and citation database. The number of entities and relationships that are available in Scopus, you can see it's 1.5 billion cited references going all the way back to 1970 when the data was starting to publish. This is what we serve to our end consumers, but to get from all the data that is collected from different journals that publish on Scopus.com. It's not just Elsevier published journals, but there are 5,000 plus publishers that publish content on Elsevier's Scopus product. To go from that point to get us to a filtered, curated, the set of numbers that you're seeing there, there's a long process to get to that. If you log into a Scopus product, you will see standard default search interfaces, which has search results. You can see also on different profiles, author profiles and institution profiles. You can see that we talk about who is written the articles, how many citations are there, what is the author's H index, who are the correlated people to this particular author. The journey from getting the data from different publications or journals into the product is a complicated journey. What problems specifically? We're not trying to solve all the problems that we had in this journey. We are looking at something specific during this pipeline. The pipeline that we had before we refactored or re-architected it, it did not enable us to make changes frequently to enrich the documents to give us better search results. Why did this happen? I will go into a bit more detail about the pipeline itself. The changes that were needed to be made on the pipeline, they were very fragile and it would break quite often. To maintain this entire end-to-end pipeline was also expensive. The hardware that you use for this pipeline was about 90 solar index, which is not actually used for live clustering. There was a Redshift cluster, and of course the data processing nodes that you use to process data between these two instances. What was the scenario with the old pipeline? Essentially, a backup copy of the index was created, data was updated in Redshift, a whole bunch of calculations across these relationships were done and added back into Redshift, and the comparison was done between Solar and Redshift, then upgraded it into Solar again, and then finally the index was copied over. So to do the traversal of the relationships and to update the index, that was the setup. So you had the previous relationships which were indexed in Solar, you had Redshift which was doing reprocessing, you were merging the results, comparing and merging and updating the results, and then copying the index. You can imagine making any change to this process would be expensive. Every time you have to make a change, you have to make changes in the Solar index, both live and the indexing pipeline, and in the Redshift schema to make sure that we calculate the right output. Looking at this particular problem, what is it that we wanted to achieve? We wanted to get a system that would perform well during runtime. Performance is important because Scopus is a live customer-facing product. Any particular one of the criteria or one of the characteristics of this problem that we were trying to solve with the metrics was every time we started, we knew what was the starting node that we wanted to start with. So if we want to do a metrics for author, we know that this is the author ID and this is where we are going to start. Most of the metrics that we were calculating were based on either depth first or breadth first traversals. All the relationships mapped together to one or many nodes, or it has to go through a hierarchy of the relationships. And the final thing was mostly we were doing matrix calculation. We were not doing full text search or full text enrichment to the document. It was calculating certain things so that you can sort or rank results differently in a search engine. That was the starting point. That was the thing that we had to solve to make our system a bit more robust. We could solve this in many different ways. You can see both Solar and Redshift are good technologies in their space. There are all viable options. You can slice this problem in many, many different ways. So why did we use a graph technology? The most important parts of the few most important things that we had for using a graph technology, because like I said, everything was a classic multi-level traversal of the relationships. It was not anything else. All our enrichment on the index was done based on data being linked to other data sets. And that's how it was enriched. There were many-to-many relationships, not just multi-level. There are many-to-many relationships. There is also non-trivial multi-level joins. That is one of the biggest benefits that you get from using a graph database. And like I said earlier, most of our enrichment on providing good search results were based on the relationship. So you have a flattened, denormalized document that you can actually use to give search results, but how do you enrich it? Most of the enrichment was based on relationship or connected data to other entities or other domains. We looked at a few technologies that were there in our landscape. Some of them were not picked up because they were overly complicated to start up with. So we eliminated few things that we deemed were not going to be possible for us to set up infrastructure wise. When we started this project, we picked two things that looked obvious and easy for us to start with: Neo4j and Neptune. Neptune, because it is available in AWS, it's hosted. The time to ramp up on it is not a lot. Neo4j being the market runner in graph technologies. We picked both of them. You can see the numbers from this is an evaluation for about more than 6 to 8 months ago. The thing that clearly marked one winner over the other was the traversal problem. The biggest difference was that we had to do complicated add-on joins from one level to another, which was difficult to do in Neptune, whereas Neo4j excelled on it. And that was the problem that we were trying to solve. Of course, there is indexing and performance and the cost of it, but that was secondary consideration. The biggest consideration was the kind of queries we were trying to resolve performed a lot better and were intuitively more suited for Neo4j. One thing I have not put up here is the data model itself. When we were doing the evaluation, we tried to use a couple of different data models and modeling it the same way in both Neo4j and in Neptune, so that we can get like-for-like comparison. They both are property stores, and we were trying to do the same kind of queries. So the comparison is like-for-like in this case. After doing the evaluation, we ended up with a fairly simple architecture. I was talking about aggregating different relationships. If you want to join this and this, the additional information that we get from the first and the second is the new link between A and C. So aggregating relationships and how we do the queries becomes quite interesting. When we push the data, if one stream of updates on Kafka is sending us this, the second stream of update on Kafka is sending us this, the output result will be this. Because we ended up with about 300K nodes in the database with a lot of relationships. We have a system that handles about 1 million updates per day. And the hardware that we used was reduced from about 90 nodes to 9 nodes. So previously, to do the recalculation, we compared it to the entire index that was existing before, and the Redshift cluster and everything else that was there. So to do the same calculation, we went from those many nodes to a smaller set of nodes just to process it. This not just simplified our pipeline, it reduced the cost, but it also opened doors for us to do other work. Like I was saying earlier, we have the relationships in the graph. You can enrich your content differently. We can add new data quite easily, and we can make new links and associations. We can do guided navigations, we can do related entity suggestions, and of course, the most obvious one was weighted ranking, the one that we started with. So we don't have to have hard metrics only to do the weighted ranking. We can calculate new metrics and provide better search results to do that kind of ranking. This is end of my talk.
I suppose just a comment or an observation, or maybe a question from me, really. You had it seemed like it was quite a substantial change in the amount of hardware that you needed to have for the solution when compared with the previous implementation on Solar and Redshift. Am I understanding that correctly?
Yes, you are understanding that. Really, that was our biggest shift. And plus the changes and maintaining that fewer number of instances is always easier, right? Solar, because we had to index the full set to do the calculations, we couldn't do it with less than 90 nodes. And every time we had to add a new relationship, the entire Solar index needed to be changed. That also meant that if you have to change your relationships, again, the whole Solar index needs to change its structure. That causes a lot of problems. Whereas with the graph database, you add a new relationship, you calculate your new metrics, and you enrich your data set in Elasticsearch or Solar, whichever one that is. That makes life easier.
Excellent, absolutely, thank you. And there's, certainly that's something I've seen with many of our other users and customers as well. And the fact that you can, you get a genuine network effect with the graph database. You can start with a given data set, and then you can add extra data sets as they become available over time and layer them in or connect them into the existing graph. And especially typically it's not a difficult thing to do, but secondly, it enhances the graph, doesn't it? It adds to the value of the graph, and the more the connections you have between data, the more valuable the graph becomes. I'd like to thank Lena and Shobhna very much today for taking the time to explain to us the very important work that they're doing with Neo4j. And I'd like to say bye-bye for now and wish everybody a great day.